Good morning and welcome back. I hope your fall semester is off to a good start. I'm Colin Wu from the Department of Chemistry, and today I'm going to talk about grant writing, and this is actually part two of my talk. Those of you who are new, I gave a short conversation about grant writing in general back in March. So thank you to Tori, Red, and Dr. Perdue for inviting me back, and I'll be happy to do this again if there's a need for it. So in part one, I mainly talked about grant writing in general, how you would search for different fellowship opportunities, really just a conversation to getting started. This particular workshop, part two, I'm going to focus specifically on the OU Provost Graduate Student Research Award, which is OU specific. So there's a bit less pressure in writing this, and my goal is that everyone here in the audience will submit one in the upcoming cycle. It's offered twice a year, and I highly encourage everyone to apply. When I surveyed around the room back in March, only about 2 or 3 students have had some experience in writing grants. And then 1 or 2 students have actually received the OU Provost Research Award before. And my goal is that the next time I were to ask this, over half the room, or in fact, I would prefer to have all the students at least have submitted an application, and maybe half of you would have gotten one at that point. Okay. And if you have any questions, feel free to contact me. My contact information is right here on the slide. And if you missed my talk back in March, I'm happy to share the slides from that presentation, or you can write an email to Tori and she has access to those slides as well. So just a quick recap of what I talked about back in March for how to get started. The main thing you should do is talk to your thesis advisor or your faculty mentor, because you really should not be writing, and as you'll see, grant writing takes a tremendous amount of time. So you really shouldn't be carving out that much time out of your research without consulting with your faculty mentor first. Once you get their blessing and they're okay with that, then you can go ahead and get started. So for this specific funding opportunity, you would go to the Oakland University Research Office website. The URL is posted here. And once you're on that website, you would navigate to the funding opportunities for students section. And these are all of the opportunities that pop up. Notice there are some that are specific for undergraduates. And then for the graduate students, these are the available funding opportunities. Some of them, if you look, are specific for travel. So this might be funding to go to a conference to present your work. The travel awards are not meant for research. Okay, so do pay attention to that. Now, the good thing about the OU funding mechanism is that it is divided based on discipline. I come from a STEM background, so my students would apply for this particular Provost Graduate Research Award, which is coupled to the science, quantitative social science, engineering, and mathematics. Students who are in music, theater, dance, the social sciences, or humanities would apply for those other categories. Okay, so it levels out the playing field a little bit so that, not everyone from engineering is getting all the research awards. It's broken down by categories. I have not checked the other links to see if the deadlines are the same, but I know the one for the STEM fields, the due date is in the middle of October, which is the main reason why I'm recording this presentation now and not trying to do a live meeting later, because I want to give you guys at least a month to work on this so that you have time to ask questions. It is offered twice a year. So if October is too soon for you, plan for applying early 22, okay? And, back in March, the larger grants, you should really be outlining what you want to do a good 6 months to a year in advance. For the OU Provost Research Award, I think a month is probably a good at the minimum sort of head start to outline your research ideas, write up a draft, and have enough time to proofread everything. Okay. And one more thing I do want to mention that these are offered, there are 2 cycles per year. And generally, the funding rates for these are fairly high. I don't want to give hard numbers. At least in my lab, students tend to have a 50-50 shot, so that if they don't get one in the October cycle, they tend to get theirs the following cycle. And the flip side is true, because it is an internal award. We do try to spread the money out a little bit. So if you were to get an award October and you try to apply again next cycle, I would say your chances would actually go down, and it's unlikely you would get 2 in a row. Okay, so keep that in mind. So if you apply this October and you don't get it, don't be discouraged. Use the comments to revise your application, submit again in the next cycle, and I would say your chances of getting it are quite high. And as for all things in life, read the directions. So if you go to, click on the link for your particular Provost Research Award, the eligibility and grant format is posted online. Follow that very closely, okay? I always joke around, and it's sort of true that scientists are just lazy in general. So we always look for excuses out of work. So if you go to even professional grant review panels, if they see deviations from the advertised instructions, your grant automatically goes into the recycle bin, right? So you don't want, it's less cutthroat, I would say, for OU, because this is meant to be a training platform. But at the same time, we do want you guys to pay attention to the advertised instructions and adhere to them, okay? So to be eligible for the Provost Research Award, you have to be enrolled as a graduate student if you're applying for the Graduate Student Award. You do need to have a GPA of 3.0 or higher. There's a little asterisk there that I'll talk about later. But we do, award these based on merit, and part of how we evaluate you is not only the science, but also you as a student. Okay. Now I'll talk about some exceptions to that later on when we talk about scoring. Okay. You of course need to have an OU faculty member to serve as your thesis mentor, or somebody who can advise and mentor you for the project. And they're going to be the ones writing you a strong letter of recommendation. So that goes back to my first point, that you really need to keep your thesis advisor in the loop of this. Don't write up something and submit it without consulting them first, because they're going to have to be involved in mentoring you, and they have to submit a letter of recommendation. And then finally, to actually submit your application, it is done through the OU internal system, which is called InfoReady Review. Access to that is linked to the grant instructions itself, so you can click on it, log in with your GRIS ID and password, and then it'll take you to InfoReady Review, where you can upload your proposal. And then the bottom line is this, right? Everyone is interested in what the award is. And so for this particular funding opportunity, it's $1,500 towards your research project. Okay, the fellowships I talked about back in March, some of these are much larger grants that actually goes to pay for your salary. Some of them even have research supply, conference travel, and equipment built in. This particular case, because it's OU specific, we don't have an infinite pot of money available. So we try to make it, enough to worth your while, but it's not going to go towards your, paying you for your summer salary or something like that, but it's going to be used for your research. Okay. And so related to that, you will need to fill out a budget form of how you will use your money. And so I would say, taking the STEM sciences, for example, or a student from my lab, they, out of the $1,500, they might use half of it towards research materials and supply, and then the other half using for consumables, things like that. Generally, what we want to see is the funds being used specifically for your project. So we don't want to see, we're going to spend $300 on gloves or something like that, because that's seen as a general consumable. But in my case, where we do more bio-related work, if there's a specific cell line or cell type that the student needs for their particular thesis project, that would be fair game. Okay, so we don't want to see $1,500 going all towards pipette tips, gloves, and things like that. We want it to be research specific. Now, I did say Conference travel is not allowed through this mechanism, but if you are in the humanities field and you need to travel or to access a database or access a particular archive, that is related to your research, and so that is okay. Okay. But if you are using the funds to travel to a conference, to network, and to present your results, there is a different funding mechanism for that. Okay, and if your research is going to involve human subjects or animals or hazardous materials, you are going to need to fill out additional paperwork. Right, so this requires, for human subject research, you're going to need IRB approval. If you're working with mice, you're going to need additional institutional approval for working with animals, so on and so forth. Now, if you look at the fine print here, and this is also posted on the instructional website, when you click on the grant mechanism, it says that you can actually apply for your grants before getting all this paperwork approved. You will need to have formal approval before you can receive funding, though. So keep that in mind. Okay, so you can still work in your grant, submit your grant by the mid-October deadline. The awards are going to be announced late November, so shortly after Thanksgiving. So you will have about a month to get all your paperwork together. Okay. All right, so now we can actually break down the grant itself. So the Provost Graduate Student Research Award, what are the different parts? Again, all of these are posted. I'm just going to go over each one in a little more detail, especially if you're new to grant writing in general. So as posted on the instructions, you need a title page. And title page, as the name suggests, is just a title of what your project is called. But there are additional hoops to jump through, right? So we want to see the applicant name, the affiliation, so meaning what college, department, or other unit you and your faculty mentor are from. And then you would include a project abstract, which is a project summary of 200 words or less. And that is key here, right? We don't want to see 500 words. We don't want to see 201 words. We want to see 200 words or less. And because your grant is going to be reviewed internally by OU faculty, we want the abstract to be mostly jargon-free. So what does that mean? What that means is that, so for example, I study DNA repair mechanisms. So there are going to be lots of names of proteins, names of techniques that we use in the lab that other people are not going to know. So to keep it jargon-free, we try to broadly describe what that technique does so that a reasonably educated person who's outside your field can still understand. Okay, so my sort of justification or the way I see it is, write it in a way where your fellow student, not particularly in your lab, but maybe somebody down the hall, can read it and understand exactly what's happening. So you want to write it in layman's terms. Okay. Now, this might not be intuitive, but I actually suggest you to write this last. The reason being, a project abstract is a highly dense, it's a 200-word summary. So in order to write this dense summary paragraph, you should have a good idea of what your overall project is about. So the best way to do that is actually outline the rest of your grant first, and maybe even write out the proposal body, and then come back and use your proposal itself to construct the abstract. Okay. So here is an example of one. So this is one that one of my students received in the February cycle. This is the undergraduate Provost Research Award, but the graduate Student Research Award has the same instructions. So as you can see here, the student has the title of their talk, DNA damage and contractile function of cardiomyocytes. I left her name out there, but the affiliation, so Department of Chemistry, Oakland University, and this would be College of Arts and Sciences, would be included in the title page. And here is her abstract. So a 200-word paragraph. Now, this is a maybe difference in philosophy and mentoring styles. I try not to nitpick on students' writing styles and grammar. I tend to read through it and make sure the overall points are outlined and that the flow between ideas is solid. And because there are infinite ways to construct a sentence, I don't want to convert a student's writing into my own style. So we're going to read through this and see if it passes through this jargon-free layman's checklist, which means to you guys in the audience who are not DNA repair experts, you should be able to read this and get a general idea of what my student is proposing to do. If not, then she could do a better job in revising this, okay? Okay. So the mechanisms behind heart failure remain largely unknown and could be greatly expanded upon. So what that's saying is that the sort of molecular details behind how heart failure occurs is not known, and we need to know more about it, right? And unlocking this information behind the disease or disease pathogenesis, so the mechanism for that disease, would allow for innovation and targeted methods for therapeutics. So knowing how the disease manifests will allow us to create targeted therapies to treat them. And so it is important to explore these concepts, explore these concepts to guide future developments in the treatment and prevention of heart disease, which is related to cardiac contractility. So the way your heart contracts, that particular function, understanding that can be tied to preventing heart disease, presumably due to irregular beating of the heart or improper beating of the heart, okay? And then in her particular case, if you read the title or go back to the title, we are interested in how DNA damage affects heart contractile function. So exploring how DNA damage affects heart contractile function could undoubtedly contribute to that particular knowledge gap. And so the relationship between oxidative stress and DNA damage will be examined to quantify the resulting profile of DNA damage on cardiomyocytes. So this is where it sounds maybe a little bit jargony and could be tightened up a little bit. So what she's trying to say is, if you look at the DNA damage profile or DNA damage distribution of cardiomyocytes, which are heart muscle cells, so that would be a more layman's way of writing it than cardiomyocytes. So looking at the DNA damage distribution of heart muscle cells, that relationship between oxidative stress and the resulting DNA damage in those cells could potentially be used as a diagnostic for heart disease. And so she is predicting that with increasing DNA damage is going to lead to a decrease in the ability of these heart muscle cells to contract. And so, overall conclusion is that these experimental outcomes would allow understanding how accumulation of DNA damage affects the progression of heart failure. So I'm sort of paraphrasing a lot of what she's saying here. The paragraph is not perfect, but I think you get an overall gist of what she is working on. Okay. All right, so that is the title page, and that is what I recommend you to work on last. The actual proposal body itself is called the project description. So for the Provost Graduate Research Award, this is capped at 4 pages, single-spaced. Even the fonts and margins are prescribed for you, so stick with those. Okay, don't try to get fancy and think you can squeeze out an extra half a page if you reduce the font to 10 or 10.5 or something like that. I know for NIH or NSF proposals, it goes through a computer system, but it automatically rejects your application if your margins and your font size and the density of the text deviates from their instructions. Okay, I don't think anybody at OU is going to look through with a magnifying glass, but first thing you should do is just set your font to Arial 11, set your margins, set your paragraph spacing before you even start typing. Okay? Your project description is broken down into multiple parts. So we will go through each of these, and you should include all of this in your project description. There is an introduction section / needs assessment. So a lot of these have little slashes on them because different fields tend to call them different things, but they all mean really the same thing. So the introduction really is an introduction of the need for your research. Your objectives are really, your goals or the aims of your proposal, significance and impact. So why is your research important? Methodology and approach is how are you going to do your experiments? Outcomes and deliverables are what you get out of doing your experiments, and then a timetable. So sort of a timeline of when you would be able to carry out your experiments. So I'm going to go back to each of those bullets, but I'm going to briefly touch on how your grants are scored and reviewed. This is also important. And I touched on this last time back in March when we talked about, the NSF fellowships or the American Heart Association research fellowships. Always look at the review criteria because that's how your grant is scored. And a higher score leads to a higher likelihood of your grant being funded. And so this is the actual rubric that faculty reviewers of the Provost Research Graduate Student Award uses to score your grant, okay? So this is from last cycle. So Dr. Madeline Bayan from the bio department recruited me to serve as a potential reviewer, which I gladly accepted. So here's me listed on there. I redacted the student's name, but look at the breakdown here, okay? So we are going to score your grant on a 20-point scale. And this is specific for this particular grant, not just all grants in general. For this particular grant that you're going to write, it is on a 20-point total possible scale. The title page is worth up to 3 points. So look at this checklist. So if your name and affiliation is on there, your project title is easy to understand, your project abstract is written in layman's terms, and it's 200 words or less, you should get all 3 points over there. Where we would deduct points is if you, left out that you're in the Department of Biological Sciences or something like that. If your project title is filled with jargon and nobody can understand it, or if you're over the word limit, we can deduct 1 point from each of those categories. But I think most of you shouldn't have problems getting the full 3 points out of that section. Now, if you look at the other chunks, your project description, which is the main body of the grant, is going to be worth the most points. And it should be, right? So the bulk of the writing is in the project description. So about half of your points, a little less than half of your points, up to 9 possible, is from the project description. Now, if you look at the other requirements, there is a short bio. So you should write a short paragraph about yourself, about your career goals, what you want to do after OU, your research interests, things like that. And this allows us to know you as a candidate a little better. And you also have to include a bibliography, so a literature cited page in the format that is traditional for your field. Okay, I recommend using a citation manager to insert those references. If you have questions about that, let me know. I can certainly refer you to some tools that are out there. But this is also really all or none, right? So if you included a bio about yourself and a correctly formatted bibliography, you will get the full 2 points. The GPA is where we had that asterisk, right? Under the eligibility section, it said you need at least a 3.0 GPA to apply. It doesn't mean that if you have a GPA less than that, you're automatically booted out, but it is a scoring criteria. It's not a huge amount, but it could make a difference. So if you meet that requirement, you will get a point here. If your GPA is less than 3, it's not the end of the world. You would just not get that 1 point. What that means is you can still get a maximum of 19 points as your total score instead of the 20. So you can still do an excellent job in all the other areas and still get your grant funded. So don't let your GPA discourage you. but it is one of the scoring criteria. Okay. As I mentioned before, you also need to include a budget justification paragraph on how you're going to use your $1,500 towards research. We don't need formal quotes or anything, but maybe a short description of what that particular line item will be used for. So let's say, $500 will be used for a, purchase of this particular cancer cell, or in the previous case, this particular heart muscle cell line that we're going to use in the lab. Another $500 in chemicals, reagents, and supplies to use in our experiments, and then another $500 on, the imaging specific materials that are tied to that project and that project only. Okay, so as long as you included a budget justification, you will get a point there. And so, excuse me. And so the sort of X factors are these two last sections, right? So I think almost everyone would get pretty close to full marks on the rest of those categories. So then you'll have 9 points here from your project description, and then 4 points here from your mentor remarks. So what your mentor remarks are is really your faculty mentors letter recommendation. Okay, so if they flat out forgot or did not send you a letter recommendation, that is almost highly detrimental to your score, because you get 0 out of 4 points, right? That's 20% of your score. If they've written a okay review, but it's clear that it's some sort of form letter that they just copied and pasted your name from a different letter, you would just get a point there. If they wrote you a very strong letter, you would earn almost the full points on that section. So that is up to your faculty mentor. In your project description, this is where you have full control of, because this is your writing. Okay, so we'll go through each of these line items, but that is what I bulleted in the previous page. Okay, so going back, so this is part of your project description. The first section is the introduction / needs assessment section. What this is really just a scientific background that justifies the need for your particular experiment. So one thing I always want to train my students on is you don't want to be doing experiments because your faculty mentor told you to. You need to think about why you're doing that experiment in the first place. And a lot of times the reasoning for that is there is a particular knowledge gap in the field, and you are coming in to address that particular need or gap. And so a general formula, and this is not, by no means the be all and end all, but this is a way to get started, is you should provide a short literature review about the field in general. So this shows that you're well-read in your field and you understand and are up to date in the current literature about your field of research. Okay, so demonstrate that you're up to date. So don't cite a paper unless, don't cite a paper that's in the 1980s, about your particular work. We'll want to see that you're up to date with, current events. Okay. Talk about the progress that has been made in your field. So, tying back to the example I showed before, where we're trying to link DNA damage and heart function. So a short scientific background is that, we know that oxidative stress contributes to DNA damage, and we can cite papers for that. And we also know that oxidative stress is a risk factor for heart disease, and we can also cite papers for that. And there's tremendous progress made in those fields in terms of developing techniques to how to study DNA damage and how we study heart health. What's missing is the merge of those two fields, right? The impact of DNA damage on heart function, that part is still not known, and that's what my student is trying to address. Okay? So demonstrate that you're up to date with your current reading, the current literature. Quickly outline the progress that's been made. It's it's a complete black box, right? There are people actively working on similar fields, but there should be a particular gap that you and only you is well suited to address. Okay, so then you want to define what that problem is. So what is that knowledge gap? So in the example case, it would be the impact of oxidative DNA damage on heart contractile function is not known. So how would we be able to address that? Well, in my lab, we have the tools to study DNA damage. We have the tools to study heart contractile function. So we're going to do both of those experiments and try to correlate those results with each other to see if there is a direct relationship, a cause and effect relationship with each other. So that's the introduction section. So notice it sets up the problem of background in the field, what is the problem, and then this next section is why is that problem important, the significance of that problem. So going back to my example case about heart contractile function, heart disease is still the #1 killer in the United States and probably worldwide. And so there's tremendous economic impacts or burden that we sink money to every year to diagnose, treat, to try to eradicate heart disease. And so understanding how this disease manifests and how DNA damage contributes to heart disease will allow us to design novel therapeutics towards that field. Okay, so why is that important? Next, and this ties into the impact and the significance of your project and the innovation of your project, is this particular statement. So assuming that you 100% complete everything that you proposed in your grants, how would that work move the needle of your field forward? Okay, so notice the answer to that question tells you how significant or how impactful your work is, right? So assuming my student is 100% successful in being able to correlate the type of DNA damage with heart contractile function observation, if we're 100% successful in that, we can potentially link defects in DNA repair directly to heart disease, right? So that has the potential impact to transform the field of how we diagnose and treat heart disease and maybe even cancer, because the two are tightly coupled to each other. So assuming you're successful in your experiments, how would that transform your field? What we don't want to see is that if you're 100% successful in your experiments, the field would have moved, zero amounts. That means there's, completing those experiments really have contributed nothing to the field, which is rarely the case, but we want to make sure you have a good justification for doing what you are proposing to do. Okay. Now, innovation can be both, or it's or it could be methodologically innovative. So the way you're doing the experiment is where the innovation is coming from. So maybe you developed a new technique to study something that could not be studied before. Or it could be conceptually innovative, or both, right? Conceptually meaning just an idea, right? So the idea of linking DNA damage and heart health, that has not been looked at before. So one can argue that would be a conceptually innovative approach. Okay. So you can address in what ways is your project innovative. And just a short paragraph to that significance section would suffice. Next is your objectives, goals, and specific aims section. Okay, so these all are synonymous to each other. So really the goals of your project. If your project is in the STEM field, a lot of times we want to see your goals feeding into a broader big picture hypothesis, right? So if you go back to early middle school, high school, where you learned about the scientific method, your hypothesis is a question, right? A question that you derive based on something that you observe. So you see something, you go, aha, based on what I saw, this is what I think is happening. So a hypothesis, a testable idea, keyword is that it has to be testable. And then your individual goals or your aims or your objectives will be the different things that you're going to do, which will contribute to testing whether your hypothesis is true or false. Okay, we'll go over examples of that in just a minute. The aims and goals themselves should not be codependent. So what we mean by that is if aim 2 is predicated on the success of aim 1, that's usually a very bad sign. Why is that bad? Well, that means if you fail in aim 1, then that means your entire project is not going to pan out. A lot of times NIH proposals, NSF proposals, this is what they, how they score your grants, right? So if they see codependent aims, that's usually a death sentence, okay? In rare exceptions, if your aims have to be codependent, there's no other way to write it that way, you can address that by showing super strong preliminary data. So maybe your aim 1 is to produce, protein X. So we're gonna express and purify protein X in my lab because protein X is involved in heart function. So we gotta make the stuff so that we can study it. And then in aim 2, we're gonna test how protein X or mutations in protein X is going to affect heart function. So look at how X affects heart function. So in this particular case, the two aims are dependent on each other. I cannot do aim 2 without having protein X in hand. So if I fail at aim 1, that means I also fail in aim 2, and that's bad. So how would I address that? Well, I can show preliminary data. So these are my two aims. If I have preliminary data that shows I have protein X made in the lab, and I have a data slide or data figure showing that, maybe on a gel or something like that, which is a technique we use in the lab to show, demonstrate we have purified proteins in hand. That is strong preliminary data suggesting that we can move forward with aim 2. Okay, so be careful with that. Now, your aims themselves should not be yes or no questions. So I put in some keywords here that that you're encouraged to use. So identify, determine, examine, characterize. So aim 1, if you have protein X in hand already, aim 1 could be express and purify protein X in the lab. Aim 2 could be identify or determine how mutations in protein X affects heart contractile function. Right, so notice that it's not a binary yes/no question that are in your goals and aims. So which means you're going to avoid words like whether, because that implies a yes or no outcome. Right, so if my aim is to determine whether protein X is involved in heart contractile function, that's yes or no. That is less interesting. So we're more interested in mechanisms of how does protein X affect heart contractile function, something that does not have a binary response. Okay. Words like explore, that's sort of in or understand, that's more like in a gray area, right? Because you can go out exploring and not get to a definitive answer, right? So we don't want you to go out on a fishing expedition and not really sure what you're going to get out of that, right? So exploring the function of protein X is not really a good way to craft that aim because it's not very focused, right? So hopefully that gives you some insight onto the phrasing a little bit. So now let's go through some example. So this particular case, I actually emailed this document to Tori, so please ask her for a copy. This is an actual Provost Graduate Student Award from a student in my lab who received this last cycle. So in his particular grant, you will see in the hypothesis section, this is what he had written. So notice your hypothesis can be a yes or no question. right? Something that's testable, right? It needs to be testable. And your hypothesis itself can be a yes/no question. So let's read through the hypothesis. Same thing, this should be jargon-free, but a reasonably educated person, in the sciences, in the biological sciences field, should be able to understand what is going on. So the way he had written is that, or up top here is what he had on his hypothesis paragraph. Differentially phosphorated tau 441 aggregates in intact cells. So there is a lot of jargon here that I don't like. But if we break down the sentence, it says that differentially, so differently phosphorylated, which is a particular modification. So, tau 441, which is a protein that he's studying. So this is just a proper noun, a name. So tau 441 is the thing that he's going to study. Differentially phosphorylated, so different forms of that protein aggregates, which is clumping together in intact cells. So that's his hypothesis, that this differentially modified form of tau 441 protein can clump together in intact cells. So that can be yes or no. You can devise experiments or goals that goes towards testing that, right? So even if you know nothing about tau 441, how would you devise a way to test whether or not differentially phosphorylated tau 441 can clump together in cells? Well, you better have a way to generate differentially phosphorylated tau protein. You better have a way to examine and confirm if they're aggregating or not. And you have to be able to do that within cells, okay? So notice your hypothesis really sets up how your aims are going to be constructed. So I've sort of rewritten the hypothesis that is slightly more in, reduce some of the jargon. There are still some keywords that, maybe somebody in the humanities won't be able to understand, but somebody in the general biological sciences should be able to deduce what is going on, okay? So the hypothesis rephrased is that the phosphorylation patterns of tau 441 protein have distinct aggregation properties in human cells. So this is just a different way of rewriting what the student has submitted. So what they're saying is that this particular modification, the patterns of that modification, so you can have different types of patterns, different modifications on that protein are going to have different properties of aggregation, of clumping together inside human cells. So that should be a easier way to understand the hypothesis. And notice that can be yes or no, right? So your hypothesis can be flat out wrong. We can do the experiments and actually find out that the phosphorylation patterns of this protein have zero effect on the aggregation properties. In that case, our hypothesis will be proven wrong. So how would we set up our aims? So notice now within our aims, we use words like to study, to assess, to determine. So these are not yes or no. But completion of each of these aims all feeds into that overall hypothesis. So going back to our hypothesis, so let me just read it again. So the phosphorylation patterns, so the modification patterns of this particular protein are going to lead to different outcomes. So different aggregation patterns, different aggregation properties within human cells. So how are we going to test that? Well, aim one, we're going to have to make tau protein, right? So in order to study this thing, we're going to have to make it in the lab. So aim one is to make the stuff, and that automatically makes aim 2 and 3 code-dependent with aim 1. But in this case, the student has shown using this gel that he has made successfully tau protein or different forms of tau protein in the lab already. Okay. So aim 2, once we have the protein in hand, is to study how tau protein interacts with other forms of tau inside a particular cell type. Okay, so aim one is to make different kinds of tau protein that have different modification patterns. Aim two is to study how these different proteins interact with each other inside the cell. And then aim three is to put everything together, right? To correlate those different modification patterns with their ability to form these aggregates. Okay, so notice by checking off each of one of these project goals, it goes back into testing the original hypothesis. So if you have questions about that, let me know. This is probably the hardest thing to write. And so one thing I would suggest is that those of you who are applying for the October cycle, start with this first, because this essentially outlines what your grant is about. You and your mentor should sit down and agree on a testable hypothesis, and then what your individual aims are towards testing that. A good number is 3, but certainly 2 or 3 aims is probably a good starting point. You don't want your hypothesis to be completed by only having 1 aim in your project, and you don't want to have 5 or 6 aims, because then that's too big for the purposes of this grant. So 2 or 3 is probably a good starting place. Okay? 2 or 3 aims for your hypothesis. The next section is your methodology and approach. So how will you actually go about carrying out your experiments that you proposed in each of the aims? Are you going to use a completely new method, or can you, reference existing methods out there that you can already utilize? So how are you going to do the experiments in the first place? And once you've done the experiments and you have data from those experiments, how will you analyze that data, right? What sample size are you going to use? How many times are you going to repeat the experiments? How are you going to assess how rigorous and reproducible your experiments are, which is now a new thing, especially if you're applying for NIH grants. We want to see that you have built-in statistical testing and ways to vouch for your own experimental reproducibility, so that if somebody else tries to replicate your experiments and gets a completely different results, that's bad, right? So you want to have additional controls built in as a way to sort of justify and interpret of what your results mean, okay? And you can speculate on what your expected outcomes are, right? So, you might see this type of data when you do that experiment. What does that data tell you? Is that consistent with your hypothesis? Or what this data that's inconsistent with your hypothesis, what would that look like, right? So what are those outcomes, and how do those outcomes play a role into figuring out whether or not your hypothesis is correct or not correct? Notice the aims themselves are not binary, but your hypothesis can be, okay? And then what are some potential pitfalls and alternative approaches? So not every experiment is perfect. And sometimes reviewers get caught up on that. And this happens in professional review panels a lot, where you propose to do an experiment using a particular method, but then you have a reviewer out there who, is zeroed in onto the weaknesses of that method, or the limitations or assumptions of that method, and they hang on to that, and their entire review ends up being negative because that's all they can focus on. So potential pitfalls and alternative approaches is a good way to address what the limitations are of your approach, right? No approach is perfect. What you can do in this section is maybe suggest what alternative approaches you can do if you see something that is unexpected. So let's say I'm going to use this new method that's highly innovative. It's going to allow me to get to the data that I need to test out my hypothesis. I have these, statistical tests built in. But then in my expected outcomes, I get this super noisy data that I can't definitively conclude what is going on. That's a huge no-no. So does that completely end my experiment? Ends my grant-funded research? Hopefully not. So if that were to happen, if this new method that's not proven yet leads to these outcomes that are not useful, do I have a different way to do the experiment to give me maybe not as innovative data, but at least get me partly there to keep the research going, right? So think about that. And then lastly, the outcomes and deliverables section is what you get out of the experiment that you put in and out of the aims that you're proposing. So I like to think of this as an input versus output, or input leading to an output. So aim one, your aims are your individual inputs, right? So in this particular student's case, his aim one was to express these different versions of tau protein constructs. So that's the input. So what is the output? Well, then the output would be those various tau proteins that he's going to express and purify. So the input is I'm going to make these constructs. The output is the proteins that we're going to use in our experiment. Second aim, we're going to study those different tau proteins and their interactions with each other in the cell. So what's the output? Well, the output is I should be able to have a quantitative description of what those interactions look like. How strong are those interactions? How many proteins are interacting? Measurable outcomes that I can get out of doing that experiment. So hopefully you can see that your input, your individual aims will lead to certain outputs, which are the results of that aim, okay? So what will you achieve by accomplishing each of your specific aims? What are those deliverables? What are those outcomes? And together, if you put all of those outcomes together, hopefully that should be sufficient information for you to test whether or not your hypothesis is correct or not. And then lastly, you do need to include a timetable. Okay, most of these projects, I would say you should be able to complete within an academic year. So maybe fall, winter semester with a summer built in, so within a calendar year. You probably shouldn't propose something that's going to take you 3 years to complete. And so you can break down the timetable by semesters. So, during the fall, I might, generate those tau constructs. In the winter semester, I'm going to collect some of this data, repeat those experiments. In the summer, where I have more time, is where I'm going to completely increase my sample size. And once all the kinks are worked out and the experimental procedures are optimized, I'm going to do this large scale data collection. And then I'm going to use the rest of the summer to write up my results in the form of a manuscript or something like that. Okay. What we want to see is feasibility, right? So we want to see that the work that you propose can be reasonably accomplished based on the time that the timetable that you're proposing and the budget that you're getting, right? So if you're proposing to do this large, genome-wide study that's going to take a team of researchers 5 years to do, chances are $1,500 and 2 semesters are not going to get you anywhere near close to that huge project, right? So the goal is for you to write something small and relatively self-contained that you can do within a year, that you can test, design an experiment to test the hypothesis, and do the experiments, analyze the results within that 1 year time frame. So with that, I wish you all the best of luck. Start early. You guys have a month if you're applying for the October cycle. Start that conversation with your faculty mentor. Let them know that you're interested in applying for this. Ask them for advice and make sure they're okay with you spending time, because it does take a lot of reading and writing time out of that. Construct your hypothesis and your aims section first. And then after that, then you can write your introduction and needs assessment, which is really just a literature review about the current state of the field, current progress of the field, what the knowledge gaps are, and then enter you into the equation of how you're going to address it. And then your approach section, which is how you're going to do the experiment. So then you can, jump around a little bit. Once you have the full document together, go back and write your abstract, okay? And of course, proofread, and make sure you spell and grammar check, right? So because this grant is being reviewed internally in OU, if your project, let's pick, something outside the sciences, if your thesis research is looking at the development of Lake Tahoe between the years 1943 and 1967, just making this up, I can guarantee you that you yourself are the only expert in that category. So other faculty, mentors, and reviewers who are looking at these grants and scoring these grants, we are not the experts. But you should write it in such a way where any reasonably educated person would be able to see what you're working on. So as long as your proposal is well organized, well outlined, and easy to read, and is jargon free and typo free, and it's written in proper grammar, you have a very strong likelihood of getting your proposal funded. Whereas if your thoughts are jumping around from sentence to sentence, your paragraphs are not making cohesive sense, there's no clear flow of ideas, it's filled with typos, grammar errors, then your proposal score is going to go way down. The ideas can still be solid, it can still be highly innovative, but if it's not written in an easy to read way, chances are it's not going to score well. So my advice is, once you have the functional draft, pass that along to another student in the lab or a colleague down the hall, have them read it, get fresh eyes on it, have your parents look it over. My parents are not science experts, but if they're able to decipher, generally what I'm working on, what the topics are, how I'm approaching it, then I think I've done a good job on conveying my thoughts to a general audience. Okay, so this part is extra important. So the reason you start early is so that you can hand off your document to fellow proofreaders so they can give you advice. If you have any questions, feel free to email me. My contact information is here. Start early. I hope all of you guys will apply for the October cycle. If you miss it, now is a good time to plan ahead for the early 2022 cycle as well. Okay, so if October is too soon for you, your classes are already picking up, you're too busying with your TA responsibilities, start thinking about for the February cycle, which is, I guess, 5 months ahead. You can still outline your hypothesis, your aims page, start writing your intro, and then you just have that much more time to polish it. Those of you who are submitting in October, you should find out in November whether you get it funded or not. So I would be happy, look forward to hearing good news from you. Let me know if you apply, and definitely let me know if you end up getting it. Hopefully this was helpful for everyone. I'll be happy to do this again and talk about other topics related to grant writing. If you guys are interested, just let Tori, Red, and Dr. Perdue know. So with that, have the great rest of your morning, and let me know if you have any questions. Thanks.